Okay, let's go ahead and get back into the yeast data. And I'm going to talk more today about principal components and introduce you to the related topic of singular value decomposition. So let me go ahead and get the data running since it takes a moment, and then we'll start talking. So I need to make this go. So let's come here and do principal components, and let's talk again about what we're doing and how to interpret the results. Okay, so once these are loaded in, the idea, again, behind principal components is that we have a matrix of numbers, call it M by N, features in the rows, samples in the columns. For this data set, for this data matrix, we could compute a sample covariance matrix. We could also compute a sample correlation matrix. Let me show you what those look like. So we have loaded in capital Y 6,740 by 20. And we're going to do principal component analysis on these data. We're going to do it in such a way that we are treating, we're basically going to be looking for a structure between the individuals, is the way we're going to do it. I have a matrix. There's rows and there's columns. I can use principal components to explore, quote, structure among the columns. Or I could ask for structure among the rows. Either way. PCA is just a visualization technique. Think of it that way. You can look at your object however you want to. First, let's think about treating the individuals. Let's look at structure between the individuals. So let's think about asking what causes one individual to be different from another. How are these individuals different from one another? They're not all the same, obviously. Here's the head of Y. Lots of different numbers. There is variation. There's differences. So thinking about the columns, these people, or these, not people, but yeast preparations, how are they different from one another? What is the story behind these data? What we do is we compute, in our case, an N by N sample covariance matrix or a sample correlation matrix. So the covariance matrix, let me just add some stuff here. Let's do capital S is the covariance matrix of Y. So we're going to have N by P. That's going to give me N by N. I think that's right. This is the sample covariance matrix. So the sample variance for column 1 is 0.08. Like variance of column 1 is 0.0838. Variance of column 2 is 0.16, and so on. And then these off diagonals are covariances between variable 1 and variable 2, covariance between variable 2 and variable 4, and so on. Okay, so think about these numbers as kind of quantifying the variability and covariability. This is a way to bundle together all the information in our data, variances and covariances. In terms of asking about structure, we're going to say, is there anything systematic about that covariance matrix? Are there component pieces of the covariance matrix that contribute a lot to what the covariance matrix looks like? Okay, so the way that we do this is we compute eigenvalues and eigenvectors. Okay, so I posted some notes on eCampus this morning on principal components. Might as well take a peek at them real quick. Notes. PCA. There's more detail in these slides than you need. They're from a different class, but you might benefit from looking through them. And in particular, you might benefit from looking through them more than once. So if you do principal components on some data, think of this set of slides as kind of a comprehensive resource for knowing what's going on behind the scenes. Let me skip to a particular slide. And we have data. We have a sample. We have that capital X M by N matrix. And we want to do principal components on it. The idea is that we have some variables. Let's see. We have variables and we have measurements on the variables. In a multivariate case, like where principal components is classically presented, we would say we have capital X, which is N by P, where N is the sample size and P is the number of variables that you have. So we might have people, and for each person, we have your IQ, we have your education, years of education, we have your annual salary, and so on. For each of N individuals, I have P variables, multivariate data. That's what the notation in the slides is assuming. You have N measurements on P variables, so capital X is N by P. We are going to try to find new variables. So think of the columns of this matrix as our variables. So it's years of education, annual salary, and so on in the columns. Let's try to take our P variables and boil them down to just one. Let's replace P variables with just one. If you're going to replace all the variables with just one, then you should pick that one in such a way that it spreads the individuals out as much as possible. Because you're trying to preserve as much information while going from lots of variables down to a smaller number. One example that I think is useful in this context is image compression. So you take a picture with your fancy digital camera, and you get a JPEG that's, I don't know, 20 megabytes on your computer or something. And you want to email it to your friend, but it takes a while to upload. Or maybe you're going to post it as a user profile image on a website, and they say can't be more than 5 megabytes or something. So what you could do is open it in an image manipulator program and say, like, export to a different format, or save as a different format, or save at a different resolution. So I'm going to take my 20 megabyte photo and turn it into, say, a 1 megabyte photo. But it better still look like me, or it's useless. So in order to go from high resolution picture down to lower resolution picture, we're doing something similar. We're saying, where is the structure in this picture? You could turn the picture into numbers, by the way. You have a bunch of cells or pixels, and you could quantify how bright and what color is each pixel. You could turn that into numbers, and it's a digital representation of the picture. So take those numbers and figure out how, like a variance or a covariance matrix, how are these numbers differing? What's the total information content? And then let's do this principal component thing to extract the most informative core pieces of this matrix. And instead of having, I don't know how many pixels there were in that enormous matrix, let's boil it down to, from 1,000,000 pixels down to, I don't know, 500 pixels. I'm taking 1,000,000 variables and I'm going to shrink it down to 500 variables. And I'm going to choose those 500 variables in such a way that they preserve as much of the original variability in the high quality image as possible. So that at the end of this process, I have a new image. It's composed of it's a smaller dimensional problem because I've boiled the variables down to a much smaller number, but it still looks like me. It might be a little fuzzy, but the most prominent features are still there because we have preserved as much of the information and as much of the structure as possible. We're kind of doing that that's what principal components kind of does. It takes multidimensional stuff and it allows you to look at it in smaller dimensions. Okay, so for looking back at those slides, we have a matrix of numbers n by p. Think of variables being in the columns, p variables. So I could compute a sample covariance matrix, p by p, or a sample correlation matrix, p by p. So this would be the variances and covariances of my variables, or correlations of my variables. The task is to take our p variables. So we have x1, x2, ... xp. Those are p variables that are being measured for each row in our data set. I want to take those p variables and boil them down to a single number, a new number, which will be a new matrix. And the way I'm going to do this is via a linear combination of the original variables. So I could do like variable 1 times the square root of variable 2 divided by the inverse cosine of variable 3 or something really funky like that. That's not linear. Linear combination is nice and simple. It's just constant times first variable plus constant times second variable plus ... That's what I mean by linear. So among all linear transformations, linear combinations of our original variables, let's try to find the one that maximizes the information content. And what does it mean to maximize the information content? I'm taking a covariance matrix, and it contains like all of our information, and I want a new variable that preserves as much information as possible. So if we wanted to separate the individuals in our data set, if I wanted to be able to separate the individuals in my data set in terms of one variable, it would be a bad idea, like I said last time, to give every person in the data set the same number, because now you can't tell them apart. You threw away all the discriminatory information. Instead, I would like to assign numbers that would make it easy to tell them apart, as easy as possible to tell them apart. So in terms of like, what would that mean? What is the goal for this new variable? It is to make its variance as big as possible. I want to take a linear combination of my original variables, and I want to pick that linear combination in such a way that it maximizes the resulting variance. So here is a linear combination of the variables. This should actually be A1 transpose X. A1 transpose X would be the linear combination of these variables. I want to find A1 such that the sample variance of the resulting new variable is highest. We're going to impose some a little restriction just to kind of fence in the range of possibilities. Doesn't affect really anything. It's just a technical thing. The main thing is this. Yes. Transpose is if I have a vector u, like 1, 3, 0, and let's say a vector v that is -1, 0, 1. So these are 3 by 1 arrays, kind of 3 rows, 1 column essentially, which is just another way of saying vector. An available operation for doing stuff with vectors is called, well, okay, so I can transpose. That's an operation. U transpose would mean anything that was a column now swings over and becomes a row. So it becomes like this. You can do that to matrices too. If you had a matrix that was n by p, then the transpose would be p by n. First row is now the first column, and so on. And if I do, say, U transpose V, that's an operation that's kind of like a multiplication of vectors. It looks like 1, 3, 0, -1, 0, 1. 1 by 3, 3 by 1. It's going to give me a 1 by 1, a scalar. And what it is 1 times -1 plus 3 times 0 plus 0 times 1. That's called the inner product of two vectors. So transpose is just a simple operation that swaps kind of the orientation. So back here, I'm looking for a vector of coefficients that I can do transpose times the collection of variables for any individual. So for row 12, suppose we have 5 variables. X12 is those 5 variables for individual 12. So give me 5 numbers, call A1 a vector of 5 numbers. Go find those 5 numbers such that when you do this inner product, this is like a linear combination. It's constant times first variable plus constant times second variable plus constant times third variable. That's a linear combination of these things. So go find A transpose such that the resulting variance is the most. That resulting variable, A1 transpose X, is called the first principal component. Principal components are new variables. You take your original ones, you boil them down to new ones, and those are called principal components. First principal component maximizes the variance of the resulting variable. Second principal component maximizes also, but now we are restricting this linear combination to be perpendicular perpendicular to the first one, which is saying the first principal component, this is the most informative way to linearly combine our variables. It wouldn't be very useful to then say, maybe the most informative way is to average all the variables. If you want to spread out the baseball players in terms of career statistics, just average all their career stats. That'll spread them out. That's the max. That'll have the most variance of all linear combinations of your variables. So what's the next best one? It could be do a slightly different weighted average, but you're still basically quantifying the same thing. It's like an index. There are two different types of index. It would be more informative to say the best direction to boil things down is in the direction of like a weighted average. And then in a totally different direction. Now that you've explained that relationship, look in a totally different direction and find out what's the next best way in that direction. So the second principal component maximizes the variance of its resulting variable, subject to it has to be perpendicular in a totally, it's telling you a different story than the first one, and so on. If you have p variables, you will have p principal components. And the hope is that you can sufficiently summarize your p variables with much fewer than p principal components, because then it makes it easier to learn something. Yes? And the first one. It would be perpendicular to everything that came from it. So here's the solution to this problem. This is the principal component problem. Solve this. Find A1, find A2, ... And the solution is to go compute the sample covariance matrix, capital S, and get the eigenvalue eigenvector pairs. Go find the eigenvalues and the eigenvectors for the sample covariance matrix. Just as a reminder, you don't have to be a linear algebra expert to benefit from principal components. The definition of a eigenvalue eigenvector pair, let's say a covariance matrix that's square and symmetric, same number of rows as columns, covariance between variable 1 and variable 3 is the same thing as the covariance between variable 3 and variable 1. It's symmetric like that. So if I can do this, if I can find, let's see, to be consistent with what we're doing elsewhere, let's find a vector A such that I can write S times A is equal to a constant lambda. That's not a vector or a matrix. That's just like 4 times A. So this is P by P. This is P by 1. This is 1 by 1. This is P by 1. If you can find A and lambda such that that is true, then lambda A is an eigenvalue eigenvector pair for S. I said last time, what is there any intuition to be gained from that definition? It's an admittedly kind of abstract concept. One thing that I find kind of useful is to say what we're doing with eigenvalues and eigenvectors is taking this matrix S and simplifying it into component pieces, independent component pieces, like that maximally capture the information in S. If you look at this definition, there is a sense in which we're taking a matrix and boiling it down to a number. So I replaced S, which was P by P, with just a single number on the right side. So there's something special about this relationship that if I know A and lambda, I can boil S down in a sense a lot. I can simplify it a lot. So it's like a structural core component of a square symmetric matrix. Eigenvalues, eigenvectors. So it turns out, if I want to find, let's say, the first principal component, well, let's just say the ith principal component. That could be I equal 1, I equal 2, whatever. The solution is to take A as the corresponding eigenvector of S. So Y a principal component is, these are the eigenvector coefficients. So E1 times X1, that should be a plus. E2 times X2 plus dot, dot, dot. Linear combination of the original variables. And I'm picking the coefficients from the eigenvectors. That is the best one. Of all linear combinations of the variables, that's going to have the highest variance. And the variance it's going to have is actually the corresponding eigenvalue. You can then say what proportion of the total variance is explained by this first principal component by looking at these eigenvalues. Enough talking. Let's actually go see it in action. So I computed covariance matrix for these data. Let's do principal components now. So let's do PC is PR comp. Okay, what do I put in here? We have capital Y, which is thousands by 20. And we are going to explore structure among the samples. We have features, we have samples. What do we want to consider as the variables? Where do I want to look for structure? Either way is a valid thing to do. What structure is there among the variables, the genes? What structure is there among the individuals? It's easier to do with the individuals because there's only 20 of them. If I did PCA on the features, I'll show you what happens in just a second. I'm going to have 6,740 principal components. And it's a little harder to interpret the results. So just as an illustration of doing PCA, let's do this first. Let's do PCA on the samples. What structure is there among the samples? What kind of structure might you expect among samples? You did a bioinformatics experiment. In our case, we have, what do our columns represent? You remind me. We're talking the yeast data. What's in a column? What? Different types of yeast. And there were different types of yeast. There are different strains. There were some that are involved in fuel production, some that are involved in wine, some that are involved in different stuff. So might you expect the genomic profile of different types of yeast to have some structure? The ones that are wine related might be expected to be similar. In terms of their genomic profile, I would not be surprised if the wine based yeast are more similar to each other than to, say, the fuel based yeast. That's what I mean by structure. Is there any kind of systematic patterns in the data? PCA is a way to explore that, to actually see, is there any structure and what does it look like? So I want to look at comparing the samples as features. Samples, treating them like they are variables. Principal components this is the way principal components is usually formulated. You have a matrix capital X, there's N rows and P columns. Samples are in the rows, variables are in the columns. Height, weight, blood pressure, and stuff are in the columns. So by default, whenever we say PR comp and pass in a matrix, R is going to say, you want to know about structure among the variables. By default, that's what PCA is defined in terms of. X is in by P. Let's look for structure among the P variables. N by P. I would get P principal components. What happens if I do PR comp Y? Let's try this real quick. That's what I want to do right now, actually. PR comp Y treats the columns of Y like the variables that we want to explore structure. So when I did this, I got some output, and I have 20 principal components. 20 principal components. Okay, so if we print just the output, PC, we see a little bit of information. We see a little bit more information if we say summary of PC. Let's look at summary of PC. Looks like that. And it's telling me the first principal component explains 55% of all the variation in the data. So I could boil my 6,740 variables down, or features down to, no, I could boil my 20 individuals down to just one and still preserve, more than half of the variation in the whole data. If you add the second one, you get an extra 10% variance explained. If you add the third one, you add an extra 8%. So to explain 95% of the data, I would need 12 principal components, for example. I'm not saying 95% is the right answer or how far you have to go, just a number I picked, 95%. So overall, looking at these numbers, it looks like there is something substantial. This 55% is a lot bigger than the next best one. There is one variable that seems to provide quite a bit more information than any other one. So there is some structure. It's not 90%. It's not like I can explain everything in terms of this, but there is a fairly substantial pattern to these data. To correlation. Think of it as taking a bunch of covariances and correlations like these. Everything is building off of that thing. I passed that capital Y, but the first thing R did was compute this. And then it's just looking at this. It's doing eigenvalues and eigenvectors of this. So this contains all the variances and the correlations. So I'm not getting back correlations here, but it's using them. It's using those correlation relationships to pick this linear combination. You can, once we're done here, you can come through and say, okay, I created a new principal component. What we could do is say, we're going to try to interpret this principal component. There is structure, apparently. What is the structure? We can learn a little bit about it. I'll show you in a second. And then you can also say, what's the correlation between this new variable and each of our original variables? So that, for example, I can explain 80% of my data with one variable, and that variable is mostly this one. So this variable right here is really important. Or maybe it's mostly a difference between these two variables, things like that. Yes? Yes. Diagonals are the variances, yes. Capital Y is median difference, or difference in median intensities between the two channels on each array. You got to have a data matrix that you start with. Then you compute a covariance matrix, and then you're ready to do PCA. We didn't have to actually compute the covariance matrix because R does it for us. You just pass in data, and it will do it. Okay, so let's look at this PC thing. When I printed out PC, what did we get? I got two pieces to the puzzle here. There was something called standard deviations, and there was something called rotation. Rotation is, I'm not going to go into why they refer to it as rotation. It's not necessary, I don't think, for our purposes. But what this contains inside the thing named rotation is the coefficients that define each of these principal components. So for example, principal component 1, this is a new variable. We're going to take our 20 variables and we're going to replace them with some new ones. And the first one, the first new column in my data set I'm considering creating, is defined by minus 0.0774 times column or individual column 1 plus minus 0.23 dot 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 times column 2 plus that times column 3 plus dot dot dot. This defines the linear combination of my 20 variables that I want. So let's do that. Let's come over here and see what happens. PC1, let's do it like this. PC1 is y m by 20. PC $ rotation first column contains the coefficients for that first linear combination. So I want to take my 6,740 by 20, and for every single row, I want to do linear combination. Take the 20 numbers in row 1 and do inner product, do that product that we were talking about, this product, where U is a row of the data and V is the coefficients that we need, those eigenvector coefficients. I can do that in one fell swoop like that. Matrix multiply m by n by an n by 1, you get an m by 1. This is going to give me one new number for every single individual. A new variable. Here's what it looks like. It's just some numbers. What's the variance of this number, I wonder? Variance of PC1, 2.022. What's the standard deviation of this, I wonder? It's just the square root of that, 1.422. 1.422. We created a new variable. It's a linear combination of the original variables. We picked that linear combination so that it would maximize the resulting variance. I want this to be as spread out as possible so that it's informative. It spreads the individuals out. Maximum way to do that is to take the eigenvector, first eigenvector of capital S, that's your linear combination. And then the proportion of variance, the variance that's going to result is, what do you think it is? What do you think this number is? If this is the eigenvector, first eigenvector of S, what do you think that is? Take a wild guess. Right, it is the variance. And in particular, it is the eigenvalue of S. We're taking S, that's like all my information, boiling it down to some magic component pieces. Eigenvalues, eigenvectors. How do you define the first principal component? It's the first eigenvector, that's the coefficients for the combination, and its variance is going to be first eigenvalue. And we can see that. Let's do this. Let's do eigendecomposition of the covariance matrix. This is going to give me eigenvalues and eigenvectors. Here's the first eigenvalue. That was the number we just computed. That's the variance of the first principal component. And notice what's in the first column from this principal component output. It was minus 0.077 and so on. I have the exact same thing here. Those are just the eigenvectors of capital S. What proportion of the variance is explained by principal component 1? I can answer that by asking the variance of principal component 1 was this. The variance of principal component 2 is that, and so on. And it turns out the total variance in the data, like let's take the sum of the diagonal elements of S. That's the total variance. Variance of individual 1 plus the variance of individual 2 plus dot, dot, dot. That is the same thing as the sum of the eigenvalues. So if I take the eigenvalues and divide by the sum of the eigenvalues, like let's do I get the proportion of variance explained by the corresponding principal component. So we can print this out. I can do eigenvalues divided by the sum of eigenvalues. And we get that. Those numbers are these numbers. Proportion explained, 55%. Independent of that story, the next best direction gives me an extra 10%, and so on. So principal components is a very handy tool. It's used a lot. It's easy to do. It's not so easy to explain. If you want to really understand what it's doing, I don't think you can get around knowing something about what an eigenvector and an eigenvalue is. You don't have to have a degree in linear algebra to understand eigenvalues and eigenvectors. They are an abstract topic, but if you read about them and look at like a Khan Academy video or something about them, you will begin to get some intuition about them. If you know what an eigenvector and eigenvalue is, you can use principal components, because that's all it's doing. So the principal, so the question, let's see if I can restate the question for the microphone. Let me just talk some. We do have these new principal components. So let's create these new principal components. They're new variables. We took our 20 variables and we boiled them down to, we might boil them down to, say, 4. 4 of them gets me nearly 80%. So I could just replace my 20 columns with these 4 variables and treat that like my data. You are then kind of limited in certain ways because they are not your original variables. PC1 involves all of the variables in a linear way. It's not easy to interpret anymore, you know. So there is definitely a penalty on like how interpretable your results would be if instead of a response variable you replaced it with some principal component. What's the effective treatment? You know, it changes this linear combination of the original variables by such and such. It's hard, you know, it can get complicated. That said, it is valid scientifically, where possible, to replace variables with something else that is an adequate replacement for them. If you had 1 million markers or something and you have some regression model for cancer, no cancer in terms of 1 million markers, and you want to predict cancer or no cancer, you could have a logistic regression model that has millions of covariates in it. Chances are, if you did principal components or something like it, you could turn those million into much fewer. So now instead of 1 million markers in my data set, I have, I don't know, 100 markers. They're not real markers anymore, though. Each marker is a combination of the millions. But if my goal is just to predict cancer versus not cancer, maybe I don't care about interpreting the things that are actually in my model. It might be that by boiling down from 1 million to 100 carefully selected new variables, I actually increase my ability to predict accurately. because some of those million are likely just noise anyway. So adding them actually hurts in terms of accuracy. Or they might be redundant, yeah. So adding redundant variables that are telling you the same thing as other variables is not going to help, in terms of accuracy. So I don't know how to answer your question exactly, because I'm not sure I understand it exactly, the nuances of it. But okay, do you have a paper? Okay. Okay, maybe there's a reason. I don't know. I'd need to read a little bit more details about what exactly they were proposing. If you have anything like an abstract or anything, I'll take a look at it. There's nothing wrong with replacing raw data with eigen stuff. You just have to, acknowledge that what you did whenever it comes time to interpreting the results. You can do t-tests between principal components and stuff like that. There's nothing wrong with doing that. Interpreting what exactly that it means is not necessarily clear. So I'll leave it at that. I'm happy to talk more. If you want to come by and sketch it out in more detail, we can talk about it. But about out of time. There's several other things that we could talk about. What else was I going to say? Oh, interpreting the principal components. Okay, real quick, let's come back up here, and the output from principal components, the guts of it is always the same no matter what software you use. It's eigenvalues, eigenvectors. Here's the first eigenvector of capital S. So what is the first principal component? We just figured out that there is something in these data. There is a way to put together in a linear way these 20 individuals, these variables, and explain 50% of the variation. That's not insubstantial. So can I interpret that story? Is there a pattern that I could actually identify? The way that you can do that, you can make something like a biplot, which I'll show you if we have time. All a biplot is doing is trying to communicate the eigenvector to you. Let's just look at the eigenvector. What is this doing? Whenever we did principal component 1, we did minus 0.077 times column 1, plus minus 0.23 times column 2, plus ... So any numbers in here that are really small, like that one, means this sixth variable isn't really contributing much to principal component 1. When it comes time to add in its contribution, it's kind of small because that number is small. Other ones, there are other small ones, and then there's several that are, reasonable in magnitude. The ones that are not further away from 0 are all kind of in the neighborhood of minus 0.3, it looks like. And there's a bunch of them. So the first principal component, if I wanted to assign some kind of meaning to it, I would say it's roughly the average of the second, third, fourth, fifth. It's a weighted average, 8th, 9th, all these rows that have numbers minus 0.25, minus 0.3. Practically speaking, whenever I do this linear combination, I'm pretty much just averaging up these individuals. And then you can ask, what does that mean? I'm averaging together individual 2, 3, 4, 5, 8, 9, 10, and so on. 2, 3, 4, 5, 8, 9, 10. 2, 3, 4, 5, 8, 9, 10. Do you see anything common? Okay, I see a couple of things. I see 3 things common, actually. Tell me one. They were all fuel except for the 5th one. They're all Stambuk, the same technician. They're all 2006. We made those box plots last time, and we saw there's a difference in 2006 versus 2005 arrays. There's a difference between the technicians. There's a difference between the fuel types. Principal components just found, just corroborated that. The biggest single source of structure in the data, 55% worth, is apparently differences, things that are specific to this collection of arrays. And it's kind of a problem because these are all fuel and Stambuk and 2006. All the fuel arrays were done by Stambuk. So are we seeing a fuel structure? Is there structure in the way that the genome looks for fuel-based arrays that's unique? Or is it just that Stambuk doesn't know how to do an array? Or whatever. That the way that she did the array was systematically different from the way that Dunn did the array. She does different amounts of reagents or something. She changes the calibration on the instrument a little bit, whatever. And or is it just a year effect that this structure is? Okay, we are out of time. I'll talk maybe a little bit more about this on Friday, but I need to show you SVD as well. If you don't have time to do the SVD, you'll have time because I'm going to tell you about it on Friday. So we'll do SVD, and I'll get you enough clustering so that you can actually do the homework, and then we'll talk more about clustering. I don't want to rush through it because it's also a handy topic. I can talk about Euclidean distance, yeah. I will do some more principal component stuff in the Q&A tonight also. So if you have more questions or you want to see more examples and more discussion like this, watch the Q&A. Okay? See you Friday.